Well, I want to thank Eliezer for actually inviting/twisting my arm to come talk to you guys, because he didn't really have to twist my arm because I didn't want to come, but more like I'm afraid because I'm not going to show you any math. I can't hide behind equations like he can. I'm a biologist, so and I'm really coming at this from a really biological standpoint. But I understand you've had a long day, and I've had a really long day too. But I can only imagine what you guys have been through, sitting and listening to lectures and talking and stuff. So I understand that, and so I apologize in advance for the number of slides and what I'm going to pack in here. If I'm not making sense, I want you to raise your hand and ask me. I'd like to save any real kind of burning philosophical questions for the end, but anything that is necessary for clarification, please do. So I basically divided my talk into two sections, which will become hopefully a little evident. It's a little arbitrary in some ways, but it's more of just a utility and practicality. So this is a slide, and in this room it may be very hard to see it, but let me just summarize the two major points from it. It basically says that psychiatric diseases are very prevalent, but they range in prevalence from things like major depression that are in the kind of 15 to 20% to schizophrenia and autism, which are hover around 1% towards the bottom. They're highly heritable, although the SNP-based heritability doesn't seem to capture all of the known heritability. And so the other point, maybe not so necessary for you guys, but I think really necessary for the field outside of of brain disorders and outside of kind of GWAS in any way, is that for a long time it was said that these psychiatric disorders were kind of garbage, right? And many of us, I'm not a psychiatrist, I'm a neurologist, but many of us, whether you're in psychiatry, neurology, what branch of medicine, know that there's real difficulty with these disorders because we don't have a measurement that we can make. It's not high blood sugar, it's not a particular bacteria or virus that we can test for. It's based on a kind of set of symptoms and signs that are seen, and often over a trajectory that one has to see as well. So to have schizophrenia, you can't just have one psychotic episode, you have to have a kind of 6-month history. So again, sometimes they're not so easy to diagnose, and different psychiatrists who are outstanding might even differ on the diagnosis of a patient quite significantly, right? We have to recognize that. But despite all of those problems, decades of research creating replicatable clinical rubric for these diagnoses, for research diagnoses, and collection of large numbers of cases have led to an increasingly large number of loci. Just here for the different disorders, it's the number of the kind of largest GWAS study. And basically the orange one, which is schizophrenia, has 37,000 patients in it. And all of a sudden, when you went from 10,000 to 30,000 patients, you had a tenfold increase, threefold increase in number of patients, tenfold increase in number of genome-wide significant loci identified. So, and one of the issues is for the other disorders, especially let's say autism, we haven't had the numbers of cases yet to really have replicatable genome-wide significant loci. So one way to think about this, and I think it's partially right and partially wrong, it just depends, is that as you increase the number of cases, you're going to get a higher and higher number of loci. There's some truth to that. But it also brings up the issue is, why now when we're almost at half this with autism and we really have no loci? What is that? Is it because of the phenotype definition, issues like that? And so we have to remain wary of that. But one lesson from this is that in general, at a first approximation, as sample sizes grow, we're finding more loci. At the same time, these are associated structural variants, which are rare, usually de novo, sometimes inherited, but very rare. And what's interesting is that despite having only about 4,000 autism cases, the number of kind of replicated significant structural variants, and now it's about double this, but at this point, was almost as many as in schizophrenia. And so the point I want to make here, and this is just shown, the different disorders here, cases in largest study, structural variants, and then GWAS here. And so this isn't in one disorder, this is just taking this data from multiple disorders and just kind of plotting it. So it doesn't really mean much, but one thing to get out of this is that, maybe the genetic architecture of disorders like autism that are severe and 30% of the time involve intellectual disability are likely to harbor more disruptive variants like de novo, protein disrupting, or structural variants, more severe mutations. And that seems to kind of be the case. And that schizophrenia and depression and ADHD, people haven't found very many structural variants or in doing sequencing, rare de novo mutations that cause those disorders. Does that make sense? So we're beginning to see, although I think it's too early to say this conclusively, that disorders like autism, we now have over 100 reasonable candidates found from exome sequencing that actually disrupt genes. And so we can actually study that gene. Whereas in schizophrenia, we don't really have any of those. What we essentially have are a lot of loci from GWAS, most of which are not associated with a particular gene ipso facto. The most GWAS signals lie in space junk, in uncharacterized, regulatory regions of the genome. They're not in protein coding. So the way I've kind of divided the talk is to talk about autism. We're going to talk mostly about stronger effect mutations and how we're looking at them, and then in schizophrenia, how we begin to annotate GWAS loci. So part one, autism. So I alluded to autism a little bit, and I'm going to speed up a little bit because I'm going slowly. So autism involves deficit in mental flexibility or the presence of repetitive and restrictive behavior in early childhood, and a problem with the social brain. And it overlaps with many other neurodevelopmental disorders, including epilepsy and intellectual disability at the clinical level. In other words, about a third of the patients have ID, and about 5 to 10%, depending on the cohort, have epilepsy. And there is a locus on every chromosome. This is old from 2008, but even at that point there were. And right now, I'm just going to summarize a decade of genetics from many groups. There are many genes predicted, probably more than 1,000 to be hit, many genetic models. In other words, you can have a recessive form of autism, a dominant, an X-linked, that's rarer, as well as the additive model, which probably accounts for about 50% of the population risk. No major gene accounts for more than 1%. So we have a heterogeneous condition where if you have 100 patients, you have 100 different forms of autism, just by definition. Does that make sense? So really the challenge is that as opposed to 10 years ago when we literally knew nothing about the genetics of autism to now, we're beginning to get a sense of real genes and some of the architecture and how to find them. And so it's really exciting in terms of gene finding. And these genes provide targets for mechanistic understanding. But from my standpoint, as somebody whose eventual goal is to translate these findings into better life for patients and their families, that means treatment, that means diagnosis, understanding trajectory. The real question is, so what? We found genes, we get high-profile papers. How do we actually move? Are we going to have to develop a specific treatment for each disorder, like 1,000 different disorders? Will there be convergence in specific pathways that then we can target? So we'll have a dozen different pathway disorders. We don't know the answer to that, but that's one of the major questions that my lab has been asking. So to put our kind of approach in context, this is just, and this is a slide from a review written by Neil Parikshak, who was a grad student in the lab, as well as Mike Kandel, but Neil made this figure. Just the idea that, when we're talking about the nervous system, we're not talking about the liver, and liver's really important, don't get me wrong. Every organ is important, okay? I'm not saying that, but the liver is pretty simple. There aren't that many cells in it, and kind of how cells are connected to each other isn't really that important. You can take a hunk of a liver out of somebody and transplant it into somebody else, and they just get a new liver. It grows back, and and they're fine. So you can't really do that with brain, and there are a lot of reasons for that, but one of them is because it's comprised of really heterogeneous cell types that are connected in regions and circuits that then lead to cognition and behavior. And so to go from genetic variation all the way to there is a tall order. And so we've just taken a really simplistic approach, starting with RNA. And we start with RNA because it's a tractable intermediate. We can measure it, it's cheap, and in fact we can measure it almost in its full dynamic range or close to that, and we can measure it in high throughput in hundreds, if not thousands of individuals. So it's something we can do. If I could do that with protein now, the same way I can do with RNA, believe me, I would be doing it. So another interesting thing is that given the role of CNV and de novo mutations, a lot of haploinsufficiency so far detected in autism, gene dosage and hence gene expression is likely to be very important. So again, just to kind of bang the model in, or in case I wasn't clear, this is a slide from Mike Kandel where you have a condition like autism, and the notion is there are different causes, some environmental, some genetic, and I left out all the heritability stuff because I wanted to get into the meat of the talk, but suffice it to say the heritability of autism was on that first slide, but it ranges somewhere between 55 and 70%. And and then the rare de novo variation, of course, will not be encompassed in the heritability estimates, except for in one, if you only depend on twins, it could skew it upwards and could be involved. And so when you take the twins out, we assume that right now about 20 to 25% of autism may be this rare de novo variation. It's at least 10 or 15%. So it's a significant component. So even the non-heritable component, let's say heritability is 70%, you can add the de novo mutations on top of that. And you can see that that genetics is contributing a lot to the predisposition to autism. But the notion here is that whatever it is, there's an altered brain transcriptome, and that is related to brain circuit dysfunction. Some of it might be proximal to this and cause brain circuit dysfunction. But over time, as we're measuring the transcriptome, we can't really know whether it's causal or caused by a consequence of brain circuit dysfunction, because we're measuring it in a 10-year-old, 11-year-old, 12-year-old who has autism after they die and we get postmortem tissue. Does that make sense? Yell out if it doesn't. So that's what we're focusing on, because we can do it. So one question is, despite this amazing etiologic heterogeneity, is there a shared pattern of pathology in postmortem brain from subjects who had autism? So if we measure the brain after the fact, we're not measuring the genes at this point, what do we find? And so just to give you some organizing principles, rather than just looking at genes that are up or down, we tend to use methods that are, that you could summarize as kind of network biology methods. And so we take the correlational structure. So if you have expression levels and you're measuring them over samples, you can group or cluster the genes based on their co-expression, from which we often infer co-regulation. We infer it, it suggests it, it doesn't prove it. Then these groupings we call modules, and within the modules you can identify hubs, and then we can try to biologically annotate the modules to figure out what they are. And we and other people, we use a method called WGCNA, which Steve Horvath and Bin Zhang developed originally. So the network structure is robust and reproducible. A gene's network position is biologically meaningful. And when I mean that structure, really the modular structure, I mean clearly if I have 100 samples and I have 10 million edges, I'm not specifying each of those edges in a reproducible way. But the modules, the 10, 20, 30 modules that you identify, those are quite reproducible. And so we can identify these groups of co-expressed genes called modules that correspond to key elements of biological function. We can identify the hub genes. So it helps us take a gene list that might be 300, 400, or 5,000 genes differentially expressed in a particular condition and organize them into something that has biological meaning, where we can infer the centrality of a gene in the process by its centrality in a module. And I'm not going to beat that more into the ground. There's some references here, and I'm happy to talk about it more. And I think maybe Eliezer gave you a few. So again, the idea is you go from thousands of genes to pairwise functional relationships. Here, it's not a PPI, it's not a protein, it's not a physical relationship. It's a statistical relationship, a correlation, mutual information, whatever you like between two genes. And so we have these groups, and so they get clustered by the strength of those interactions. And within that, we can identify modules, which is somewhat arbitrary. And so they just get color-coded. They get named colors or given numbers. And then we can ask, okay, well, let's annotate those modules. But we go from kind of, again, 20, 30,000 genes to tens of modules. And so you could ask, where are the genes of interest? For example, if I know now 100 candidate genes for autism, where do they lie? Are they randomly distributed across this? Or are they in a particular module that has specific biological function? And so that would just be an example of clustering in a module. And then you want to understand what the module does, of course. And so this is where it gets interesting and also very difficult. You want to understand what that module really means. So we want to look at how the modules, let's say first principal component or eigengene, really reflects space and time, physical interactions, and gene regulation. So for example, if we have the first principal component or eigengene, we can look over time in human brain development and see what's the trajectory. Is that a module whose genes are expressed during fetal neurogenesis, or is that a module expressed during aging? We can look at how it's expressed in regions, and now with cell type specific data, we can begin to ask what kind of cell, is that representing a particular type of cell or circuit?
Is the network from autism patients looking only at the differentially expressed genes from normal patients, or are you looking at a network for normal development?
So far, I haven't specified that, so your confusion is warranted. It could be either. I could do it in either way, and so I'm going to do it both ways. The first way, I'm going to show you what the network, very quickly because it's published, what it looks like if we just compare autism brain to control brain and the network preservation. The second part, I'll go through something more like this. But I just wanted to give a sense of kind of like what we're trying to get to, what one could do. So this is just a cartoon just to give you another confusing example. So when Irina Vonagu, who was a postdoc in the lab and is now in Sydney at University of New South Wales, she took about 20 autism brain, cerebral cortex, 2 regions, and cerebellum. These are all regions that had been implicated in the disorder in some way or another. The postmortem, ages from 4 to about 52, took the tissue, ground it up, did microarray gene expression, and asked, were there any modules associated with autism? And again, this was published in Nature in 2011, so I refer you to it. I'm just going to summarize it. I'm not going to go through all the details. But she found 2 modules that are seen in normal brain to some extent, but that were strongly associated with autism. One, and this is the first principal component, showing that it's down in autism. It's called M12, and this is M16, which is up. If we look at the gene ontology for these, it's involved in synaptic function and neuronal function, and this was up in inflammation. Interestingly, we had done a previous study showing that there was a reproducible cellular transcriptome from the cerebral cortex of humans, and this module that was annotated by gene ontology as inflammatory immune turns out to really correspond to the normal activated human microglia and astrocytes. And we could only know that because we had the modules from brain. It just points to, there's a huge deficiency, as many of you know who use gene ontology, and this was one area where it really was really weak. It was not super helpful. So like if we just graph the top 300 connections, the top 300 edges, and the nodes associated with them, this is ASDM12, and it has a number of interesting genes, including a splicing factor, RBFOX1, that's neuronal specific. And so Irina went on to show that splicing was dysregulated, and actually the targets of this splicing factor were dysregulated in proportion to the downregulation of that splicing factor in autism brain using RNA-seq. And then when she actually looked in these 2 modules, we have no causal, it's hard to make a causal inference here. We can say these modules define the molecular pathology of autism. They're seen in about 3/4 of the cases. So that was a striking thing, in that we have 20 cases of autism. We have to assume that they're due to 20 different genetic or environmental causes, because no single gene causes more than 1%. Most are really rare. And yet we have this shared pattern in about 3/4. And so she asked, well, now if I take GWAS data or I take sequencing data, and use these modules as kind of pathways, can I actually test, can I see an enrichment? It kind of gives you a causal anchor. If you found that GWAS signal for autism was enriched in one of these modules, it might tell you, it could tell you that there is a possibility that there's genetic variance that is causing these changes. Of course, we'd have to connect those to EQTL, which would be the next step. And actually, what she found is that this module was quite enriched for autism GWAS, even though it's quite underpowered even at the time. But this one was not. That's just shown here. This is M12. This is the permuted enrichment, and this is the actual enrichment. And this is what happens with M16. The P is close to 1, actually. So the model is that, we have neuronal gene dysfunction during development, and that leads to some synaptic dysfunction. And the microglia and astrocytes are upregulated in response to that, but are a critical component of that. And in fact, microglia in the brain aren't just inflammatory cells. They're involved in synaptic development and synaptic homeostasis. So we think that this is really a synaptic dysfunction that we're seeing. So we're kind of defining the molecular pathology of the disorder. So we've now gone on, and we is the royal we, Neil, Vivek Swarup, Grant Bellegarde, and Emily Wu have gone on to now replicate this in a much larger sample set, close to 100 cases and controls now. This is a little old, including 8 patients who have a single monogenic form of autism called 15q duplication that's almost 90% penetrant for autism. So it's one of the most penetrant single gene forms of autism, or single gene, major gene, because it's a duplication of a large chromosomal region. So we took 4 major approaches: differential gene expression, WGCNA, differential splicing analysis, and microRNA. I'm not going to talk about the microRNA today. I'm only going to touch on some of these things really quickly. But one of the main things that we see, and many of you do RNA-seq probably know this, is that the major source of variance, the first principal component in any RNA-seq data set are all the Picard tool metrics. And so you have to take into account all of the sequencing statistics, which are taken into account here, as well, because we have postmortem brain, is the RNA integrity. It's really, really critical. So, in fact, when we look at the first principal component, Mike Kandel has done this. He's downloaded tons of data from the web on microarray data sets on Affymetrix, where you can see 5 to 3' degradation. The first principal component is more correlated with that than with any other feature. So we spend a lot of time normalizing and worrying about that. So if we just look at the discovery set, there's the first principal component, there's the replication set, there's the controls. We're able to replicate this both in the RNA-seq, and then this is just showing how that first principal component, once we've taken care of all the potential confounders, is what it's associated with. Slight association with age, but the major association is diagnosis. And again, we can look in other data sets and see that essentially this is a fairly specific pattern. Although it is interesting, microglia are also upregulated in Alzheimer's brain and other conditions. It's not specific to autism, as you'll see later. And we do see that signal. We can also look at alternative splicing. There are a lot of ways to do that. We used a program called MATS that was developed here. And what's interesting is if you're a neurobiologist, these are known autism susceptibility genes that harbor de novo mutations or copy number variants, but they're critically involved, all of them, in early neural development, fetal brain development, which is a harbinger of something to come. If we again cluster based on the splicing changes seen in all the subjects, we get a non-random clustering. Again, about 2/3 of the cases, almost 70%, kind of are in this cluster with autism, red. And what's interesting is, again, it involves a lot of the same gene ontology components affected by differential expression, but not the same genes. And that was pretty interesting. It's also neuronal specific. These are not glial splicing events. So the other thing that we looked at are these, this monogenic form. We were very curious about it. And you can see by its clustering here in blue, these are the duplication 15q patients, that they share a very strong signature. And actually, the signature really distinguishes them from controls even more strongly than autism. And it actually encompasses this autism. So if we look at the autism versus control and the DUP15 versus control, we see that there's a very high correlation, but that the slope is 2, indicative of the fact that these changes are greater, whether they're down or up, in 15q duplication than in autism. Perhaps because it's more homogeneous, perhaps because they're more severe. We see essentially the same thing with the splicing changes if we look at the percent spliced in using MADS. So one of the critical questions is differential expression causing splicing? How are these related to each other? One way to get at that, and it's a tough question that really has to be experimentally addressed. But one thing that Neil Parikshak did was just basically look at the protein coding genes, the non-coding RNA, and the differential splicing, taking the first principal component. I didn't show you the long non-coding RNA, but again, there are about 64 long non-coding RNA that are differentially expressed. And included in that are a number of primate-specific long non-coding RNAs. But you can see a really high correlation between all of these kind of different measures. And this, to me, indicates that we're kind of looking at a unitary process from different angles. When we look at splicing, we see splicing is dysregulated, and it's dysregulated to about the same extent in the same patients as long non-coding RNAs and expression. Does that make sense? So we kind of have this unitary process. I can't prove to you that it's causing autism, and there's a lot more interesting data in here that I'm happy to discuss with anybody because this story goes on and on and on and is in re-review now. But we were able to actually do some experiments in vitro in neural stem cells to actually validate some of the relationships that we see here. And so that's pretty exciting. But at this point, the way that we look at this is we don't look at this as a pattern of gene expression that is necessarily related to the causes of autism, because multiple causes end there. It's a convergent pathway that probably represents this whole misadventure, the developmental misadventure of the brain when a person is born with the risk for autism, and then the brain experiences and develops over time, over a 3 to 5 year period, and you end up with abnormality in circuits. And that is represented by these transcriptional profiles. In other words, we kind of view this as both probably containing adaptive and maladaptive features of the brain that are responding to having autism. But it is pretty striking that this really heterogeneous, diverse, genetically, ideologically diverse condition that's pretty, that's not defined that clearly, behaviorally. It's tough, unless you're really trained. Comes down to at least in 2/3 of patients, to kind of a shared molecular pathology. Does that make sense to people? Okay, to some degree, that's enough?
Clarification question.
Sure.
How many of these modules did you say there are in total?
Well, in that particular data set, there are about 18, roughly, 18 to 20. And so 2 of them were strongly associated with autism. Now, in the RNA-seq data, it's around the same number. It's around 24 or so. And because the RNA, when we did a WGCNA, we used unweighted WGCNA. Now we've used weighted so that the negative and positives are separated. And by having more samples, we see the same pattern overall, but the M12 is now divided into 2 modules that are going in opposite directions, and the same thing with the M16, but essentially the same. So now we have 5 autism modules out of about 25.
What evidence here points to developmental issues, specifically, I guess, normal?
I haven't shown you anything that really points to that yet. Really, there's some hints, right? Developmental genes are involved. Developmental genes are spliced. I didn't show you a whole piece of the story where we actually show that there's a developmental patterning defect in autism when we compare frontal and temporal lobe. But the data that I'm going to show you a little later will get to this much more clearly. But one of the things we found was that if you look at normal cases, controls, there are about 500 genes differential between frontal and temporal lobe. In autism, there are about 50. And we're able to find some of the drivers of that and actually experimentally validate them. But that's neither here nor there. You asked, so I'm telling you. I was trying to kind of keep it a little simple. But here what we have is just the notion of convergence in that over development, at some point, there is a kind of shared molecular pathology in the majority of autism. But so far, it's just kind of more suggestive. Yeah. So one of the questions is, how specific is this transcriptome? Do you see this in other disorders? So Mike Kandel, who is a fellow in the lab, postdoc, and a psychiatry resident at the same time, has spent the last 18 months downloading microarray data over actually thousands of microarrays, but now, once it's cleaned, to about 900 or so samples using microarray, and went through a very, very careful quality control to throw out outliers and to batch normalize and batch correct. And so we did differential expression analysis looking at the global transcriptome, and he also ran network analysis to see if he could identify modules that were unique or shared across the disorders. And so there he is having fun. So what you can see here is that the transcriptome correlation, Spearman-Rowe, is highest for schizophrenia and bipolar. Next, autism, schizophrenia, autism, bipolar, schizophrenia, major depression, et cetera. And that some of these, like autism and alcoholism or bipolar and alcoholism, are negative. So it's not just postmortem brain that's driving this. We also used inflammatory bowel disease as well as a kind of outgroup, which has inflammation but isn't brain. So what's interesting here in some ways is that this somewhat very loosely seemed to parallel the shared heritability that's been published between these disorders using SNP-based heritability measures. So we asked the question, is it related to shared heritability? Is the shared transcriptome, the extent of transcriptome correlation, related to shared SNPs? And so here, he took the microarray data and now has downloaded a number of other RNA-seq data sets that have become available since February, and taken the genetic SNP-based correlation right from the PGC data that's published, and with the transcriptome correlation either in microarray in red and blue in RNA-seq, and it's very high. So at least a significant proportion of the variance in gene expression, we think, can be explained by shared heritability. And we can break this down into modules and find modules that are shared. And the beauty of that is this just tells us at a global level, let's say, there's an R of 0.56. The question is, well, what is responsible for the sharing biologically? So the modules correspond to genes that are co-expressed, and hence, we infer that they're co-regulated, involved in common biological processes. And so you can look and see that there are these different modules with different colors now that can be annotated using gene ontology. Again, I'm not a huge fan of this, although we do it as part of our knee-jerk. This turns out to be, somewhat informative in that this that's upregulated is called immune response, defense response. This is the microglial module that we find. This is another glial module, yellow, that's also up in autism and in schizophrenia and in bipolar, and that actually corresponds to astrocytes, which is another glial inflammatory cell. The other downregulated ones shown here and there are actually neuronal modules. So again, we can begin to separate this process and look at the actual biological components of the transcriptome sharing. And again, autism most similar to schizophrenia, but schizophrenia and bipolar do not have the upregulation, very, very strong upregulation of microglia that's seen in autism. At least this would predict that. So I just wanted to give you a taste of how we can begin to kind of use these transcriptomes as an intermediate quantitative phenotype to begin to look in a way that isn't, let's say, biased by my measurement of some aspect of a patient's cognition or something, to really look at shared and distinct biological vulnerabilities. Again, does that make sense? So, again, more to the story, happy to fill it in, but the real point here is that one can use genomic features as a kind of intermediate phenotypes to look across the disorders, and this indicates the transcriptome is likely to be quite useful. So the next question gets to, I'm sorry, Mr. Hat with blue water bottle. Do autism risk genes converge on any developmental time point or biological processes? So here we want to get into causality because, again, the transcriptome is super interesting, but it's always a little unsettling, like when we're making causal inferences about it. We try to do experiments, but we can't make really strong statements about what's causing what. So this is work, again, that was led by Neil Parikshak, who was a grad student in the lab. That's his everyday lab attire down there in the bottom right. The lab is definitely quite formal. So the idea here is that you can take genes that are different forms of mutations, and again, the red arrow there is just like the effect, a cartoon showing the effect size of mutation. So like red would be, major gene, probably sufficient to cause a disorder, and the other smaller ones down below are the common SNPs. And so the question is, you can ask, once you start to get a big enough basket, do these genes and do the genes implicated by genetic variation in a causal way converge? And so here, one basically creates a network that looks unbelievably like the one that I showed before in that cartoon, but actually this network comes from developmental data. So what Neil did is he took RNA-seq data from the BrainSpan resource, which from 8 weeks to 3 years of age, from the cerebral cortex, multiple individuals at each stage, and ran WGCNA. And so we're looking at normal brain development and asking, if we have risk genes, where do they fall in place and time? So the idea is, are autism mutations highly enriched in specific modules? And then we can annotate those molecular pathways. And it turns out, just to give you a summary in case you fall asleep, transcription, or haven't fallen asleep yet, but will, transcriptional regulation, chromatin modification, and neurogenesis are really where these genes sit in different modules. And then we can explore the module relationship to time and cell type, which I'll show you. And I'll just give you a little flavor of how Neil did this. So here is these are 3 modules, and this is birth, shown here in red. Time is on the x-axis, and relative expression of the first principal component on the y. So you can see that this is a gene that, this is when most cortical neurons are being born. This is the beginning of the birth of the cerebral cortex, and this is the birth of the person. And so here one can see that these are genes that begin to be expressed after neurons are born, kind of, and keep going up after birth. They're kind of later. This is a little bit earlier, and this is, perhaps kind of intermediate. And they're all synaptic or cell adhesion, and these genes are involved in the development of neurons and neurogenesis and the regulation of early synaptic plasticity. And so it turns out that if we take a whole bunch of genes that are candidates in this, we had to do something unbiased, and even though this is not satisfying, the GWAS in autism, as I mentioned to you, there's very little that's been identified convincingly and replicated. So what we have to kind of use there is we use hits from a few GWAS that aren't replicated, and then we use candidate genes that have been studied over the years that are in the Safari database that are probable autism candidates. And when we look at those, it's mostly inherited risk, and it falls into synaptic development. And what's interesting is all of these modules are super enriched in these genes that are downregulated in the adult, in the autism postmortem brain, so adolescent and adult. And so these are modules that kind of come on during mid-gestation as neurons are being born. Now if we look at de novo risk, and this is a much more interesting category for many reasons. One of them is because these genes are they're derived from exome sequencing studies that, although underpowered in some ways, we can identify recurrently hit mutations that have a high probability of being true autism risk genes. And so we can take those that are protein disrupting, which have the highest signal, missense mutations, protein disrupting and missense together. And of course, we can look in the siblings where I'm going to tell you there's no signal. And we can also look at synonymous mutations as a kind of background. So now if we look at the main modules, I mentioned to you that 17, 16, and 13 were enriched in what looked like more like inherited variation weakly. If we look, it's a totally different picture for these de novo mutations. There's really significant enrichment, especially for the protein disrupting these two M2 and M3. And again, it's a pretty small sample size, but this is all we had. And again, if we look at the trajectory of these modules, these are expressed at very high levels at the peak of cortical neurogenesis, then are downregulated and are super enriched for transcription factors and chromatin modifiers. And again, we can look and we can see that these modules are super enriched for protein-protein interactions. In other words, they're representing true kind of biological processes and protein interactions that are ongoing during neural development. And we discussed some of these pathways, such as BAF complex and other chromatin remodeling complexes that are almost certainly involved in neurogenesis. And so this would tell us that the de novo large effect genes are really enriched and targeting more likely to hit early mid-fetal development. We also took a list, just for your interest, of genes that are known to cause intellectual disability, Mendelian forms, of about 400 genes. And those were not enriched in any of these modules. They're spread out all over development and all over function. So the other thing we can do is we can begin to ask, okay, if we have these different modules, some that are up high and then come down during development, others that are going up, are they co-regulated? And we can look in the 5' untranslated region of these genes and identify enrichment for transcription factor binding sites, which then can be validated by chip experiments, which were done in some cases shown down here. And we can see that there are a bunch of transcription factors that link these processes. In fact, these MEF transcription factors have been known to be mutated in autism now, so they're great candidates, as well as SATB1 is involved in neurogenesis, as well as FOXO1 in neuronal polarity. So these are really great candidate genes. And the MEF set are kind of validated by chip chromatin IP experiments. And we can also take there are a lot of things we can do with these modules. I just wanted to give you a few examples. One of them is we can ask, okay, are they expressed in any particular, now we know that the M2 and M3 that contain these large effect de novo variants are expressed during early fetal development. Are they involving any specific types of neurons? And to make a long story short, as we get more and more single cell profiles, or we can begin to ask about enrichment. And what we find here, I'm just going to summarize this, is that the risk genes are enriched in the superficial layers of the cortex and the glutamatergic projection neurons. Why do you care? What does that mean? Those are the regions that connect the cerebral hemispheres to each other and connect cortical regions to each other. They're much less likely to project down and exhibit like motor function. So they're kind of involved in higher cortical function, and it's the regions that are most expanded in primates. So that's pretty interesting because there's all kinds of imaging and cognitive data that those circuits are disrupted. So this would fit with that. So our kind of model, and again, just a model, is that ASD genes from multiple sources converge on prenatal neurodevelopmental processes. If we take the rare de novo exome variants in ASD versus no ASD siblings, we can see that those red are enriched in transcriptional regulation early in brain development, but that these genes that are differentially co-expressed in the postmortem autism brain, or that are candidate genes, are upregulated. And the ID intellectual disability genes show much less specificity. So another thing that you can ask is, well, now that we keep sequencing, would these modules have kind of predicted anything? And there was another paper that was published right alongside this in Cell in 2013, and if you look at the modules they identified, none of those are really replicated. You can't reproduce them. And there are a couple reasons for that I'm happy to discuss. But as this new sequencing occurs, we still see that these de novo protein disrupting mutations are enriched. These are two different papers and two different cohorts still remain significantly enriched in these modules. So we think it's some real biology. We created a browsable network for biologists so that people can go in and link. And you can also link to all of the metadata behind every single point. You can see what the edge values are. You can see where all the data came from. And their instructions. So now, in the next 10 minutes, do I have 10 minutes, or am I supposed to finish? Or Eliezer is?
Yeah, you have 10 minutes.
Okay, I'll just go really fast. I'll just get to the chase. So I'm showing you how, if we have a really complex neuropsychiatric disorder, we can begin to start to organize really a myriad of diverse genetic hits and begin to try to pull the biology together. Another problem that we see is that as we start getting SNP data, how do you assign SNPs that are outside of genes to function, especially in unannotated regions? So this just highlights those issues. And Haejung Won in the lab asked, can understanding of chromatin structure and gene regulation be used to identify this? And again, we didn't focus on autism here because there's almost nothing there at the GWAS level. But in schizophrenia, there's really a remarkable paper in 2014 with over 100 loci associated with schizophrenia. But only in 10 instances was the association attributable to a non-synonymous exonic polymorphism. So how do you assign them? Again, this is Haejung, bad picture, and collaborated with Jason Stein and Luis de la Torre, who aided 2 other postdocs in the lab. So usually what happens is you find the nearest genes, or you take all the genes within the LD block. That's usually a ton of genes, or a pathway set gene analysis once you find these. So we asked, can we use high-C or chromatin contact information to help with this? So you're all, I'm sure, familiar with ENCODE, and what ENCODE has been doing has also been remarkable in that it's annotating the functional elements of the genome by saying this is a likely enhancer in a particular tissue. However, it does not necessarily attach that to a gene. People usually attach it to the closest gene. But the question is it really regulating that closest gene? High-C, I'll show you in the next slide, it will become clearer. What you basically do, so what we did is we went to our favorite organ, the brain, during fetal human development. We took the area that has the developing neurons called the cortical plate, and then we took the germinal zone, which has all the dividing progenitors, separated them from 3 different individuals in duplicate, so 12 high-C libraries made by Haejung. What you basically do is, you cross-link the DNA, and if 2 regions are interacting really close to each other, they will get cross-linked, and then you pull them down with a biotin, and they're digested, biotinylated, ligated together, and sequenced. And so you get these 2 pieces of DNA that might have been 700 kb apart next to each other, so you can see that they were actually interacting. You get a read that includes both of them, a short read. Does that make sense? And so this is actually a beautiful way, and Job Decker and Bing Ren have shown that this is actually feasible. It's really a technical tour de force. So Haejung did this in this fetal human brain and then asked, well, if schizophrenia is in part a neurodevelopmental disorder, maybe this can inform some of our understanding of schizophrenia. But first, we had to do a bunch of validation, and maybe I'll just say that, just to keep it short, we found the same kind of relationships of our physical chromatin interactions with genomic features that others had found as well. So we had no mysteries and no surprises from that standpoint. Haejung did this at a high enough resolution using in situ high-C. So we have about 10 kb maximum resolution. That's enough to identify enhancer promoter loops. So to kind of validate things first, she just used data that was out there, DNA hypersensitivity correlation from fetal brain that was in the roadmap. And you can see there's a very significant relationship between if something is interacting by high-C and the correlation of the DHS peak to a transcription start site of the gene. We can also use, using another sequencing technique. There's data from CAGE sequencing, from the CAGE consortium in Japan, where they actually have sequenced the enhancer RNAs that are coming off as the RNA is being transcribed. And so you can look at the correlation between the active enhancer and the expression of the gene. And again, that's significantly shifted so that if something is interacting by high C, it's much more likely to be interacting at the correlational level. She showed the same thing with the EQTL interaction pairs. This is just showing over different distances up to 100 kb, the log2 normalized contact frequency from high C, and we can see that if we look at the interaction between the EQTLs and the contacts, that it's most in cortical plate and less in ES cells. So there's a kind of tissue-specific regulation that's really important. Of interest, about 40% of the interactions that we see in cortex, you see in fibroblasts and IMR90 or published ES cell data. So it's not like all the interactions are cell-specific. It's that these short-range looping interactions, about half of them look like they're tissue-specific. So then her question was, let's try to take the schizophrenia loci and annotate them. So the first step was to try to kind of identify some region or some area containing causal variants. And to do this, she used a caviar which Eliezer's group developed. And so the basic Bill understand the high C interactions for each distance using all the imputed SNPs, run caviar to get the causal variants, obtain the high C interactions. And we do it within 1 megabase because people have shown that 1 megabase is where most of the action is. And we selected the interacting loci that pass an FDR of 0.01, and we assigned genes interacting to the schizophrenia GWAS SNP. So if we find a physical interaction between the region containing the SNP and the promoter of the gene, we assign that SNP to the gene. And so this is the basic, one of the main take-home messages. If you would just use the closest gene, as was used in the original paper and most people use, or genes in linkage disequilibrium, you do not identify many of the genes of action, and whether you use cortical plate or germinal zone. So here, this is just showing that these are the interactions that are occurring in high C. These are those that are occurring in genes that are in LD as well. So about 2/3 of the genes that are in LD show a high C interaction that's identified, but not all of them. But the real issue is that a lot of stuff is being missed. And what it's really showing is that the mean distance for these interactions is in the hundreds of kb. So another question that Jung asked was, well, okay, if this is really something, and if these GWAS SNPs are really doing something and misregulating candidate genes, then maybe they'll be dysregulated in schizophrenia brain. So if you just take the genes from linkage disequilibrium from the original study only, or the closest gene only, there's no enrichment over background. But if we take the caviar or the PGC credible SNPs, either of them, those that are identified in a tissue-specific manner by high C show a very significant enrichment. So just to look at what some of this data looks like, this is the LD block, this is the kind of original SNP, and you can see that it's interacting with a gene several hundred kb away. This is the negative log 10 p-value on the y-axis. This is the topologic association domain, which is the area in which most interactions actually occur. These don't change much between cells. They're kind of like relatively fixed. But these gene loops do. And so this is showing that this is not interacting with that or that, but that. SOX2 is a gene involved in neurogenesis. This is really interesting. This is the dopamine D2 receptor, which was a huge candidate gene because the drugs that treat schizophrenia act on that receptor. The SNPs are 20 kb outside the gene, and there's the LD block, and we can now show that these SNPs actually do interact with the promoter of that gene. Another interesting thing now, if we take the genes that we identify using the high C intersection with the schizophrenia GWAS, Jung finds that the acetylcholine receptor activity is the top gene ontology category pulled out. That category of genes was not identified in the original GWAS, and it wouldn't have been because not all these are the closest genes. And again, I can just show you examples of those. There's a cluster of cholinergic receptors, and this shows that this is interacting with that particular receptor. Here, it's not the closest gene. There's a very, very gene-dense region, but it's actually interacting with this SNP region, interacting with that promoter region. So again, I can go through this and show you numerous cool examples, but this is a really interesting one. This SNP, this is another gene that we were interested in because it also regulates neurogenesis. So there are a whole bunch of genes here that regulate neurogenesis, and that previously had not been implicated in schizophrenia. So Jung was really interested in this because it's about 700 kb away from the index SNP. So she was really interested in saying, well, can I actually experimentally validate this? So, yeah?
How do you know it is a FOXX gene instead of the RP11 in this case?
Well, at this resolution, you can't, but you look at the region in the genome. I mean, this is a huge area. This is 700 kb of the genome browser, so you have to look carefully. So it's not, it is interacting with that gene and not with that gene. Yeah. It's just because of the resolution of this graph, it's hard to see. But you don't know for sure. It's just a hypothesis anyway. You know that there's a physical interaction, but you don't actually know that it's regulating it. So one way to do this is we took, Jung took human neural progenitors, and she did a couple things. One is she just did a luciferase assay in non-neural cells to show that the SNP actually did regulate a reporter in the direction expected. But if she CRISPRs out the locus, about 500 base pairs or 760 base pairs around the locus using 2 different CRISPR constructs, she gets a very significant decrease in gene expression and not a change in gene expression of the closest gene. So this is experimental validation, and she's now working on SOX2 and has preliminary data that, which is about 400 kb away, is also being regulated by a SNP 400 kb away. So I guess one of the bottom lines here is that we not only want the kind of linear information on the chromosome, we need tissue-specific biological, hopefully cell-type-specific information on the actual chromatin interactions that are ongoing. The three-dimensional structure is quite powerful. So in conclusion, gene network approaches provide an organizing principle for understanding molecular phenotypes in the brain, and we can capitalize on genetic findings using convergent evidence from transcriptional networks to begin to understand the neural system's basis of a complex psychiatric disorder. We can see the shared molecular pathology. We can use this to look cross-disorder. And we can show that ASD risk genes converge during early fetal brain development in glutamatergic neurons. And we can use these findings, if we know this, and we know that the modules are conserved in vitro, we can begin to use this to model these various mutations in an appropriate system to begin to identify convergent molecular pathways. And I think with the last example, I just gave you a taste, and I'm sorry for going through it so quickly, but we have the integrative genomic tools in hand that should provide an unbiased context for understanding the function of genetic variation. I think we have to move from kind of just expediency, deciding, well, I'll just put it in the closest gene. My GWAS looks really cool. I have 100 loci. To actually using other data that's out there to, to functionally annotate. So with that, I will close. It's really hot. You must be tired. I mentioned all these people as we went along, but Neil Parikshak, Mike Kandel, Vivek, Jung Won, who really led the last part of the work. We collaborate with Jason Ernst, who's keeping us honest, as well as Eliezer. I have a long-term collaboration with Steve Horvath and Giovanni Coppola with all the network biology. This is the lab. See how happy they are? Oh, actually, there's somebody, even a disembodied person, coming by Skype. And then we use the BrainSpan data, which is this great resource. Again, another shout out for producing resources and making them available, as well as a long-term collaboration with Shyam Prabhakar at GIS in Singapore on stuff that I didn't talk about today, as well as Ben Blanco at University of Toronto on the splicing changes in autism. And the work Shyam's doing is trying to connect this to epigenetic mechanisms. Anyway, thanks for your attention.